What if I told you that a cancer cell doesn't actually spend years and years mutating just to learn how to spread?
Right. It's a crazy thought.
Yeah, what if instead it actually possesses the superpower to metastasize right from the very beginning of its journey? Today, we're basically throwing out the old textbook on cancer progression.
We really are. I mean, it changes everything we thought we knew. Classically, the medical field has always viewed metastasis, the spreading of cancer to other organs, as this very late stage event.
Like, it takes a long time to happen.
Exactly. The traditional thought process is that a tumor grows, it slowly accumulates more and more genetic mutations over years, and eventually, if it gets lucky, it unlocks the ability to spread. But the research we're diving into today completely shatters that timeline.
And we are doing a custom-tailored deep dive for you today, looking at a groundbreaking April 2026 study from the journal Nature.
It's a fantastic paper.
It really is. We're looking specifically at colorectal cancer, trying to solve this core mystery of exactly when, where, and most importantly, how a tumor acquires the ability to move. Because, according to this data, the biological superpowers needed for metastasis, they appear right at the starting line.
What's fascinating here is just how early we're talking about. To understand it, we need to focus on what oncologists call T1 stage colorectal cancer.
Okay, let's unpack this, because T1 sounds like ground zero, but what does that actually look like physically inside the body?
So think of the lining of your gut like a layered defensive wall, kind of like a castle.
Okay, a castle wall.
Right. T1 represents the absolute earliest stage of invasion. The tumor has initially grown in the superficial topmost layer, but in T1, it is just barely broken through the very first barrier.
Which is what exactly?
It's a thin muscular lining called the muscularis mucosae, and it's just entered the underlying tissue layer, which we call the submucosa.
So the castle wall has literally just been breached by the invaders.
Exactly. And that newly breached border, that exact front line where the tumor is pushing into the new healthy tissue, is what we call the invasive front. That is where all the action is happening.
Okay, so we're zooming in on that exact microscopic battle line. And the paper uses this massive term to describe what the cancer cells are doing at that front line. They call it oncofetal plasticity.
Yeah, it's a mouthful.
It really is. And if you're listening to this and wondering what on earth a fetus or an embryo has to do with colon cancer, you're not alone. I was wondering the exact same thing when I read it. What is actually happening to these cells?
Well, let's break the word down. Onco refers to the cancer. Fetal refers to an embryonic developmental state.
Right.
And plasticity just means the ability to change shape and function. It's malleable. So oncofetal plasticity means these adult cancer cells are essentially reverting to a primitive, highly adaptable embryonic state.
Wait, but why would a tumor want to act like an embryo? That seems like it's going backwards.
Because embryos are the ultimate survivors. Embryonic cells are specifically designed to grow rapidly, adapt to new environments, and literally migrate through the body to form organs.
Oh, wow.
Yeah, so by turning on these old regenerative gene programs that are normally only active when a fetus is growing, or, when your body is desperately trying to heal a major wound, the cancer cell becomes incredibly resilient.
Ah, I see. It's like an invading army stripping off their bulky, highly specific, standard-issue uniforms and putting on specialized, lightweight survival gear.
That's a great way to put it.
They drop the rigid identity of a colon cell so they can slip through the mud, enter the bloodstream, and survive the journey to set up camp in a completely different organ, like the liver.
That is a perfect analogy. They shed their mature identity for survival. But to actually see this invisible wardrobe change happening, the researchers couldn't just use a normal microscope.
You're right. They needed something more advanced.
Exactly. They used a technology called spatial transcriptomics, specifically tools like nanostring geomics and whole transcriptome panels.
Okay, let me make sure I'm picturing this right, because the paper gets pretty dense with the tech here. Is spatial transcriptomics essentially like a highly detailed Google map of the tissue, but combined with a live feed of what the cells are actually doing?
Yes, that's exactly it.
So it doesn't just show you where the houses are, it shows you which houses have their lights turned on and exactly what kind of lights they are.
Spot on. Instead of just grinding up the tumor and looking at all the genetics in a blender, which is what older sequencing did, and it loses all the location data, this tool lets them look at a slice of the tumor and see exactly which genes are turned on in exact geographical coordinates.
So they can see the different neighborhoods?
Precisely. They mapped out the normal tissue, the adenoma, which is the precancerous area, the dense core of the tumor, and that critical invasive front.
And what did they find when they looked at the map of these early T1 tumors?
They mapped 19 of these early tumors, and the data was startling. The cells sitting deep inside the tumor core were just acting like standard cancer cells.
Just doing their normal tumor thing.
Right. But the cells sitting right at the edge at the invasive front had their lights flashing wildly. They were expressing specific genes associated with fetal states and wound healing. Genes with names like MMP7, LAMC2, and ANXA1.
LAMC2, that's one they mentioned a lot, right?
Yes, LAMC2 is a major marker for the state. Furthermore, they found these cells were expressing what are called high relapse cell, or HRC, signatures.
High relapse cell signatures.
Yeah, these are gene programs that scientists previously thought only existed in late-stage, deadly, metastatic cancers.
So the superpower is already turned on at the border right at the beginning.
Exactly.
But here's where I started getting really tripped up by the data, because the researchers didn't just stop at those 19 tumors.
No, they went much wider.
They looked at a massive tissue database of over 230 early T1 tumors, tracing the patients over 5 years to see who actually got sick again. And they found these dangerous, shape-shifting fetal signatures in the invasive fronts of almost all the early tumors.
Right.
Even in the patients who survived and never had their cancer spread.
It creates a massive biological paradox.
It totally does. If I'm understanding this correctly, we've got a tumor that already has the superpower to spread at the earliest possible stage in almost every single patient.
Yes.
But clinically, we know that only about 10% of Q1 tumors actually end up metastasizing.
That's the clinical reality, yeah.
Okay, let's just do a quick recap here for you listening to lock in what we've covered in these first 5 minutes.
Good idea.
Bullet 1, metastasis superpowers, this oncofetal plasticity. They appear very early at the T1 stage.
Check.
Bullet 2, the cells express regenerative or fetal genes, like that LAMC2 marker, right at the invasive front.
Exactly right.
And bullet 3, having the power doesn't mean it's successfully used, as seen in the 90% of patients who don't relapse. So, if almost all these early tumors have cells capable of spreading, why do only about 10% of them actually do it?
That is the million-dollar question.
Hold that thought, because the environment holds the key. But before we get to the environment, researchers had to figure out why the cells at the border were putting on the survival gear in the first place. Did they just get a new, unlucky mutation that gave them this shape-shifting power?
That's the most logical hypothesis. Nature right. To test if this shape-shifting was hardwired into the DNA, the researchers performed an incredibly elegant experiment.
I love how they did this.
Yeah, they took what are called punch biopsies. These are tiny physical core samples, and they took them from 16 different patients across different zones of the tumor.
So they sampled the different neighborhoods again.
Right. They took a punch from the normal tissue, one from the precancerous adenoma, one from the tumor core, and one right from the invasive front. And from those samples, they grew 73 different living 3D models in the lab, which we call organoid lines.
Just to help you visualize this, if you're listening, an organoid isn't just a flat layer of cells in a petri dish, right?
No, not at all.
It's a 3D, living, biological avatar of the patient's specific tumor region. It actually grows and forms complex structures in a dish.
Precisely. And because they are grown directly from the patient's own tissue punches, they preserve the exact genetic background of that specific tumor zone.
That's so smart.
It is. So they grew all these organoid lines, and then they performed whole genome sequencing, or WGS, on them. They were hunting for a genetic culprit. A specific DNA mutation that existed only in the invasive front organoids.
Something that could explain why those cells, and only those cells, were shapeshifting.
Exactly. And they found the usual suspects, of course.
The classic driver mutations for colon cancer.
Yeah, things like APC, GP53, KRAS, and SMA4 loss.
The heavy hitters.
Right. And this is where the entire genetic hypothesis just completely falls apart. When they compared the DNA of the invasive front organoids directly to the tumor core organoids from the exact same patient,
Yeah.
there were absolutely no extra driver mutations.
Wait, hold on. I need to stop you there. If the core and the invasive front are genetically identical, how can one be actively preparing to spread while the other just sits there acting like a normal tumor?
It's wild, isn't it?
That breaks everything I thought I knew about cancer mutating to survive. Are you telling me their DNA is exactly the same?
Identical. The karyotypes, which are the overall patterns of their chromosomes, were a perfect match. There was no unique metastasis mutation hiding in the DNA of the invasive front cells.
Okay, so let's look at what this actually means. If we think about the cell like a computer, The hardware, the DNA is exactly the same, whether it's in the middle of a tumor or at the bleeding edge.
Exactly.
That means this oncofetal plasticity, the shape-shifting ability, it's not a hardwired genetic trait. It's more like a temporary software program being run.
That's a beautiful analogy.
It's an acquired state driven by something entirely outside the cell. The tumor cells aren't deciding to shapeshift on their own. They're being told to.
It's not nature, it's nurture. And if we connect this to the bigger picture, it fundamentally changes how we view cancer progression. The tumor isn't just randomly mutating its way to metastasis in a vacuum.
Right.
It is actively reacting to its surroundings.
Let's do another quick recap for you because we're about 10 minutes into this deep dive.
Let's do it.
Bullet 1. Scientists built 3D living models, those organoids, from different parts of the early tumors.
Yep.
Bullet 2. Genetic sequencing proved the invasive front cells are genetically identical to the core tumor cells.
There are new mutations.
And bullet 3. The shapeshifting is non-genetic. It has to be triggered by external signals. Which leads to our next big question. If the tumor cell's DNA isn't pulling the strings, who is whispering in the tumor's ear at the invasive front?
To find the whisperer, we have to look at the neighborhood. In oncology, we call this the tumor microenvironment, or TME.
The environment around the tumor.
Right. If the cancer cell's the seed, the microenvironment is the soil. If it's not the tumor cells driving the change, it's their neighbors.
So how did they investigate the neighbors?
They utilized single-cell RNA sequencing and a massive spatial map, specifically a tool called CosMx, to profile over 1.25 million individual cells.
1.25 million. That is an insane amount of data.
It's a huge map of the neighborhood. And they were looking for the prime suspects.
Which turned out to be fibroblasts, right?
Exactly. Fibroblasts.
But normally, fibroblasts are the good guys. They're the structural architects of our tissue. They build the scaffolding that holds our organs together.
Under normal conditions, absolutely. But cancer is highly manipulative. In the presence of a growing tumor, these local builders become corrupted.
Oh, man.
Yeah, they turn into what we call cancer-associated fibroblasts, or CAFs. And what the spatial mapping revealed was this beautiful stereotypic patterning that mimics the normal gut. It wasn't random chaos.
Like different zoning laws in a city.
Exactly. In the densely packed tumor core, you find one specific subtype of corrupted fibroblasts. They're called telecyte-like CAFs, and they express genes like SOX6 and BMP4.
Okay, a telecyte-like in the core.
But when you look at the invasive front down in the submucosa where the castle wall was breached, the neighborhood completely changes. Here, you exclusively find a totally different subtype of corrupted fibroblast known as trophocyte-like CFs.
Trophocyte-like. A good way I've heard to remember that is tropho sounds like the bottom or the trench. These trophocyte-like CFs are the ones down in the trenches at the invasive front.
I love that mnemonic. The trench workers. And they express different markers like SFRP2, DREAM1, and FAP. But here is the really crazy visual mapping correlation.
The smoking gun?
The smoking gun. These trench-dwelling trophocyte-like CFs perfectly colocalize with our dangerous, shape-shifting LEMC2 positive tumor cells.
Colocalize meaning they sit right next to each other?
Exactly. They are sitting right next to each other, practically holding hands, exclusively at the border.
But where did these specific corrupted CAFs come from? Did the tumor bring them along for the ride as it invaded, or did they migrate from somewhere else?
Neither, actually. And the way they figured this out is fascinating. They used a computational algorithm called trajectory inference, specifically pseudo-time analysis.
Wait, pseudo-time? How do you get a timeline or a time-lapse if you only took one single biopsy snapshot of the tissue?
It's incredibly clever. Imagine you have 100 photographs of a person from birth to old age, but they're all mixed up in a pile.
Okay, totally out of order.
Right. Even without knowing the dates on the photos, you could arrange them by the person's physical aging features to recreate their life timeline.
Oh, that makes sense.
The algorithm does that with cells. By looking at thousands of cells at slightly different microscopic stages of gene expression, it can infer the evolutionary timeline of how one cell type morphs into another.
That is wild. And what did the timeline show for these fibroblasts?
It showed that these corrupted CAFs were actually derived from the normal, peaceful tissue builders, the resident trophocytes that already lived in the submucosa.
No.
Yes. As the tumor physically breached the mucosal wall and pushed into their territory, the normal local cells were corrupted and transitioned into these trench CAFs.
It's literally like a peaceful village being invaded, and the local structural engineers are suddenly drafted, brainwashed, and forced to build siege engines for the enemy.
That is exactly what happens.
Let's do recap number 3 for you guys, hitting the 15-minute mark.
Perfect timing.
Bullet 1, the tumor microenvironment is highly organized, like zones in a city.
Yep.
Bullet 2, corrupted structural cells called trophocyte-like CCAFs exist exclusively at the invasive front.
The trench workers.
And bullet 3, these CCAFs completely surround the dangerous shape-shifting cancer cells. So we have the suspect at the scene of the crime, but proximity isn't proof. How exactly are these fibroblasts forcing the cancer cells to change?
You're right to be skeptical. To prove causality, the researchers had to recreate the crime scene in the laboratory.
How did they do that?
They did this functional test using a 3D co-culture experiment. They took those pure tumor organoids we talked about earlier, the ones with no extra mutations, and grew them together with the fibroblasts.
Okay.
They embedded them in a dense 3D matrigel setup to perfectly mimic the physical, squishy environment of the invasive front.
So they essentially built a microscopic terrarium to see how they would interact.
Exactly. And the mechanism they found was striking. When grown alone, the tumor organoids stayed relatively normal. But when co-cultured in 3D with the fibroblasts, the tumor organoids strongly induced those oncofetal HRC signatures.
The fibroblasts were actively flipping the switch.
Yes, they were actively inducing the shape-shifting.
But how? I mean, they don't share a nervous system. How are the fibroblasts communicating this to the tumor?
Through what's called a paracrine conversation. Paracrine signaling is basically cells passing chemical messengers to their immediate neighbors.
Like whispering across the fence.
Right. And by analyzing the signaling gradients, the researchers actually decoded the specific words being spoken by the fibroblasts. It turned out to be a specific chemical cocktail.
What was in the cocktail?
First, the CAFs secrete a potent signaling protein called TGF-beta, specifically PGF-B3.
PGF-beta. I've seen that pop up in a lot of immunology papers. It's a major regulator.
It is. And the second part of the cocktail involves an enzyme. The CAFs express an enzyme called PTGS2, which is more commonly known as COX2.
Wait, COX2, isn't that what everyday painkillers like Advil or Celebrex target?
Yes, exactly.
Yeah.
TOX2 produces inflammatory molecules called prostaglandins. So the corrupted fibroblasts were constantly bathing the tumor cells in a highly inflammatory soup of TGF-beta and prostaglandins.
We really want to talk about how they proved this, because the paper details this glowing green experiment using CRISPR, and it is just brilliant. Can you explain?
Oh, the validation experiment is beautiful. They used CRISPR to put a fluorescent glowing tag on a specific oncofetal marker gene called EMP1 in the organoids.
What?
Right. It's a known driver of the fetal state. The setup was simple. If the tumor cell turns into the dangerous state, the gene turns on, and the cell literally glows green under the microscope.
Like a biological burglar alarm.
Exactly.
Yeah.
So they have these dark, quiet organoids. They bathe them in normal signals, nothing. But the moment they drop in that specific cocktail of TGF-beta and prostaglandins,
They light up.
the organoids literally light up green. It's definitive proof. The corrupted fibroblasts are the coaches shouting the instructions, and the tumor cells are just executing the play.
That is just incredible. Okay, so we solved the mechanism, but this brings me right back to the beginning. I'm stuck on this paradox.
The 10% clinical reality.
Yes. If almost all early T1 tumors have these CFs, and they're all pumping out TGF-beta and prostaglandins, and they all get these shape-shifting cells, why do only some actually spread?
Because having the survival gear to travel isn't enough. You also need an invisibility cloak.
An invisibility cloak.
Yes. The source data provides one final, elegant puzzle piece, the immune evasion bottleneck. The researchers went back and compared the rare metastatic T1 tumors against the non-metastatic ones.
And what was the difference?
The immune system. The metastatic ones showed a massive downregulation of immune-related expression programs, specifically CD8 T-cell signatures.
For you listening who might not be immunology nerds, CD8 T cells are basically the assassins of your immune system, right?
Yeah.
Their job is to patrol and eliminate corrupted cells.
Precisely. The CCAFs give the tumor cells the survival gear, the plasticity, to detach and live in the bloodstream. But to actually escape, the tumor also needs an invisibility cloak to hide from those CD8 T cells.
Wow.
If you have the gear but no cloak, the immune system spots you and destroys you before you can spread.
So it requires a perfect, terrible storm. Metastasis fails without both.
Exactly right.
So if I'm synthesizing this entire journey for you listening, the story of early cancer spread isn't about a rogue cell randomly mutating. It's an environmental story. When the tumor pushes into the submucosa, normal trophocytes become corrupted CCAFs. These CCAFs release TGF-beta and prostaglandins, basically acting as coaches that train the tumor cells to become highly adaptable, fetal-like survivors.
That's the mechanism in a nutshell.
But the final hurdle to actual metastasis is escaping the immune system.
And this raises an important question, something that fundamentally shifts how we might treat cancer.
What's that?
If the tumor's ability to spread relies on a chemical conversation with its neighbors, could the future of treatment involve simply blocking that conversation?
Oh, wow.
Yeah, perhaps targeting the fibroblasts or the TGF-beta signals directly, rather than just trying to kill the cancer cells with toxic chemo. We could just jam the signal.
That is a completely different paradigm. Well, we want to thank you for diving deep with us today. As you go about your week, maybe ponder how your own microenvironments, the people, the media, your physical spaces might be silently shaping your own outcomes and adaptations.
It's a very similar concept.
It really is. Are the signals around you helping you grow or corrupting your potential? Keep questioning, keep digging, and we'll see you on the next deep dive.